Hello everyone, welcome to May 2025 Biochemistry session INI-CET recall. Don't stumble over something which is behind you. Let's go, grow and move on. So this is the first message I want to give you before starting this recall session. So let us see the questions. Analyzing the question, what I could understand is the total around 15 questions were from biochemistry. Out of that, the difficulty level is definitely it's a moderate to difficult level, and the topics which was covered maximum was definitely from genetics. Around 3 to 5 questions were from genetics. Then comes carbohydrates, lipids, amino acids, proteins, and one question from acid base balance. So most of the questions, if you ask me how many previous year question was there, direct repeat was there. That was only one question. That was a question on that Curie's disease. Then rest 9 to 10 questions were from previous year topics only. That is why I am saying moderate to difficult difficulty level. I'm saying not so easy, but moderate to difficult. The new questions came was which I have seen is one of the question that is from Eye of Tiger sign, then the gene pool, then surface plasmon resonance and strain typing. These were the new questions added into the list of INI-CET. Okay, so now we'll see the questions one by one. Question number one is a 4-year-old child presented with developmental regression, hepatomegaly, cherry red spot. The startle reflex is normal. Which of the following lysosomal enzyme is deficient here? So before analyzing the answer, let me tell you how to approach such questions. So when you see this question, first thing that should strike to you is some unique feature of that any metabolic disorder. So that is a clue that is given here. It is a cherry red spot is there, which means that this is diverting our attention towards sphingolipidosis. Now our aim is to differentiate between various sphingolipidosis. And even if you don't know whether it is sphingolipidosis, see the options. It is all something related to sphingolipidosis. So that is how we are going to analyze this question. I'll give you a simple algorithm to diagnose a case of sphingolipidosis. As cherry red spot is given in the question, we shall start with cherry red spot itself. Divide it into three. Absent or plus or minus is there or present. So that plus or minus, I'll mark it first itself, that is Krabs disease. Sometimes it can be present or sometimes it is not. So we'll go to first what is given in the question is cherry red spot is present. So once cherry red spot if you're seeing, then you have to check whether there is visceromegaly. So if there is visceromegaly or not. So visceromegaly is absent in certain conditions. Which are they? It is one is the Tay-Sachs disease, which is considered under the GM2 gangliosidosis. Then metachromatic leukodystrophy, visceromegaly is absent. Then Krebs disease, that is also visceromegaly is absent, but cherry red spot is present. Now, visceromegaly present. Visceromegaly present, it is Niemann-Pick's disease, or it can be a case of GM1 gangliosidosis. So that is one side. Now, if the cherry red spot is absent, if it is given there is no cherry red spot, then you are confined only to two. That is Gaucher's disease and Fabry's disease. That is how we learn my girlfriend has no cherry red spot. The girlfriend stands for G and F. That is Gaucher's disease and Fabry's disease. So if it is in the absent, then what you have to check for is the massive visceromegaly. Massive visceromegaly is something that you can find in Gaucher's disease, but not in Fabry's disease. So, if you want to find out whether it is Fabry's, you have to check for other clues as well, like Maltese cross appearance, angiokeratoma. If such things are given, it is a case of Fabry's disease. So, this is how you generalize and find out what is a sphingolipidosis. Okay, now in this case, as cherry red spot is given, I have started with cherry red spot. But there is one more option that you can do. That is, you can also start with mental retardation. So, if there is mental retardation given here or not. So, if there is no mental retardation, that is mental retardation absent. So, mental retardation absent, then also it is going towards Gaucher's and Fabry's disease. So, these are two ways whereby we can find out various sphingolipidosis. Now will go to that question. So there is a cherry red spot present in the macula. And what is given here is there is developmental regression. Hepatomegaly is present. So this is something with the cherry red spot. See, with the cherry red spot, and there is visceromegaly. So that is confining us to sphingolipidosis. That is Niemann-Pick disease and GM1 gangliosidosis. Now we will see the option. So we are shortlisted to these two. Now we will see the option. Hexosaminidase A. That is definitely, we know it is Tay-Sachs disease. In Tay-Sachs disease, there is no visceromegaly. Sphingomyelinase, yes, that is Niemann-Pick disease. That is the answer here. Then aryl sulfatase A. Aryl sulfatase A is metachromatic leukodystrophy. That is not having visceromegaly. Glucocerebrosidase. That is a case of Gaucher's disease. Gaucher's disease, two things are against here. One is cherry red spot. It is not present. Number two, visceromegaly. That is present. That is favoring Gaucher's disease. But the cherry red spot is absent there. So visceromegaly is present, but cherry red spot is not present in the case of Gaucher's disease. So definitely that is also not the answer. So here the answer is sphingolipidosis. So this is a simple algorithm I would advise you to follow whenever you get a question on sphingolipidosis. Next question is gene pool refers to all alleles in. Gene pool means that it is a pool of like the genetic material that we are collecting from different alleles in a population. Why? Because we want to guarantee something that is called genetic diversity. So if we want to like guarantee, or if we want to make that pool which is having genetic diversity, it should be from a population. If we are taking an inbreeding gene pool or different alleles which is taken from an inbreeding population, then we won't get that genetic diversity. Whereas if we are taking that from a what you call as a population, then we can guarantee genetic diversity. Okay, so that is what is given according to National Genome Research Institute glossary. It is given the definition of a gene pool. It is a combination of all genes, including all the alleles presenting in the reproducing population or species. So then only the genetic diversity it can be guaranteed. Whereas if we are taking an inbreeding population, then it is having less genetic diversity when it is compared to a population where you can see it is a reproducing population or species. There can be genetic diversity. So the answer here is definitely it is not from a family. The genetic diversity is not there. Not from an individual, definitely genetic diversity is not there. And not from a tissue also. So it is from a population. Then another question, a very straightforward question. Which of the following is correct? I think in among this molecular biology technique, this is the easiest molecular biology technique question that was asked. So we all know snow drop. So this we will cancel. So S is for D, N is for R, and W is for P. So what is the answer here? N R S D W P. Yes, A is the answer here. Next is neonate was normal at birth, developed liver disease, muscle weakness 4 months later, severe hypoglycemia is there, particularly in the morning. It says that it is fasting hypoglycemia is there, hepatomegaly is there, so it is pointing towards a glycogen storage disorder. Ketosis is there, then acidosis is there, so ketosis definitely causes pH reduced, then increased ALT and AST. Then what is given as administering IV glucagon. So following a carbohydrate meal, a normal rise in blood glucose level is seen. This is the most important clue that is making us or helping us to make a diagnosis. A normal rise in the blood glucose when we are giving an IV glucagon challenge after a meal. Whereas overnight fasting, we can't differentiate between von Gerks and Cories based on overnight because they are giving the same result. Okay, so what is it? So I'll come to the answer later. So here, definitely the most probable answer is Curie's disease, where a debranching enzyme is defective. So that is alpha 1414 glucan transferase. That is what is defective here. So, what are the factors that favoring of diagnosis of Curie's disease? Definitely fasting hypoglycemia. Then hepatomegaly. That is common for all glycogen storage disorder, most of the glycogen storage disorder. Then another thing that is favoring for Curie's disease is increased liver enzymes. That is favoring Curie's disease. But there are features of type 1 von Gerk's disease also in the same question. That is, one, ketosis. Ketosis is mostly seen in one, but three also sometimes may or may not. But ketosis is a biochemical hallmark of type 1. So that is sometimes may mislead us to a diagnosis of von Gerk's disease. Glycogen store. It is not mentioned whether it is normal glycogen or abnormal glycogen. So if it is given abnormal glycogen, then it is more towards Curie's disease. If it is given normal glycogen, then it is more towards von Gert's disease. Yes. So it is given glycogen. So we have to take it as a normal glycogen. That is also something which is against our Curie's disease diagnosis. So there are features which are distracting us from the diagnosis of Curie's disease here. Some features are there. At the same time, certain features are favoring Curie's disease. So in such condition, what we do is a challenge or a test which is called as IV glucagon challenge. So IV glucagon challenge is we can do it, that is we are giving a dose of glucagon two times. One is after meals and during, this is not after, during overnight fast. These are the two situations where we are giving IV glucagon. But during overnight fast, we can't diagnose anything. So I'm not discussing that. Now we'll come to after meals. So, or with meals, if we are giving glucagon, we know that if we are the patient is having food, which means that there is a blood glucose level already in the blood because the patient is taking food. So there is a baseline blood glucose level already there. Now on the top of that, what we are giving is glucagon. What's the role of glucagon? It accelerates or favor glycogenolysis. Okay, let it favor. Now we are suspecting two conditions here. Either it is Curie's disease or von Gert's disease. Now, what will happen in von Gert's disease? In von Gert's disease, we know the deficiency is in the glucose 6 phosphatase. So even if we give glucagon, the final enzyme is not there. So the blood glucose level will not rise. Whereas if it is Curie's disease, then we know that only debranching enzyme is defective. So till all the branching points, some of the glucose 1 phosphate can be released by glycogen phosphorylase. This glucose 1 phosphate can be converted to glucose. So will there be rise in the blood glucose? Yes, there is a rise in blood glucose. It means that this is a case of Curie's disease. So the answer here is debranching enzyme. And again, I should say one thing. This is an exact repeat question that was asked in an AIMS, not I think in 2018 or 17. AIMS question, the same question came. So that is only one question which I am seeing as a previous year question, direct repeat. Next is how many cycles of PCR is required to generate half the copies of 26 cycles. So how will you find out? We know that if X is the number of amplicon, then 2 to the power of n is the formula by which we calculate the number of amplicons, which means that if X is the number of amplicons, then the number of the nth cycle or the nth cycle number, we are getting X number of DNA. Let it be anything. X number of DNAs we are getting. So that is it. Now the question here is, when are we going to get X by 2? So the x is calculated by the formula 2 to the power of n, which means here the question is asking 2 to the power of n divided by 2, when are we going to get? So we know that 2 to the power of n divided by 2 is 2 to the power of n minus 1, which means that the cycle where we are getting half the number of that DNA is n minus 1. For example, in this case, it is given 26 cycle, and half the number we are getting at cycle called as 26 minus 1, which is 25. So if this is complicated, I'll tell you a simple example to learn the same thing. We know that after cycle 1, how many DNAs we are getting? 2. After cycle 2, how many DNAs we are getting? 2 to the power of 2, that is 4. Okay. After cycle 3, how many DNAs we are getting? 2 to the power of 3, that is 8. So just see the relationship between all these things. See, just prior to that. So if this is the second cycle, the first cycle is having half its number. If this is the third cycle, and just prior cycle is having half its number. Okay. So if you know that this is 2, 4, 8, it is going in geometric progression. 2 to the power of n, it is exponential progression, which means that half the cycle is just one cycle before. Okay, so that is about which cycle. So answer here is definitely 25. Next question is an obese individual with a weight of 90 kg, high carbohydrate diet, which enzyme is responsible, or which enzyme is prominent or most responsible for weight gain? Okay, so we are given many enzymes. It's a multiple response question. So we'll see what happens if a patient or person is on high carb diet. High carb diet, definitely it is splitting into glucose. This glucose, some will be stored as glycogen. Okay. The next, it undergoing glycolysis. Then pyruvate is formed. If there is excess glucose taken, we know it is going into a valve, and that is called pyruvate dehydrogenase. This reaction, where is it occurring? Inside the mitochondria. So once it enter into this valve, it can't go back, which means this acetyl coenzyme A, which is inside the mitochondria, is actually has to enter into some other pathway. So for that, this acetyl coenzyme A has to be transported, because it can enter into fatty acid synthesis. Fatty acid synthesis is not taking place inside the mitochondria. Then where is it taking place? It is inside the cytoplasm, which means this acetyl coenzyme A has to be transported. So to transport this, it is transported as a 6-carbon compound, that is called citrate. And this, it is transported as citrate, and it is transported by which transporter? Tricarboxylic acid transporter. So there is a role for tricarboxylic acid transporter. Now once it get into the cytoplasm, this acetyl coenzyme A is converted to malonyl coenzyme A. What is the role of malonyl coenzyme A here? This malonyl coenzyme A is taking a major role. One, it is going towards fatty acid synthesis. So that is given here. It goes towards the fatty acid synthesis, and it is stored as triacylglycerol. Okay. And this enzyme which convert acetyl coenzyme A to malonyl coenzyme A is the rate limiting enzyme of fatty acid synthesis, and that is acetyl coenzyme A carboxylase. Okay, fine. Then is there any role for malonyl coenzyme A apart from this? Malonyl coenzyme A is a inhibitor of one gateway of beta oxidation. That means if fatty acid synthesis is taking place, it is going to put a brake on fatty acid oxidation. How? Malonyl coenzyme A is closing the gateway of beta oxidation. Which is the gateway of beta oxidation? It is CPT1. So malonyl coenzyme A is also playing a major role here. So now we will see the question. Malonyl coenzyme A, yes. Acetyl coenzyme A carboxylase, yes. And mitochondrial carnitine, no. This is for fatty acid oxidation. And I've told you malonyl coenzyme A is blocking the CPT1 gateway, which is the transport system. Then tricarboxylate transporter, yes. So one then 2 and 4. These are all answers. So the answer here is 1, 2 and 4. So if from these options I will take only one, I will take 1, 2 and 4. But I have a suggestion for you. That is, if the option were 1, 2, 4 and one of the option was 2 only, I will take 2 only as answer, not 1, 2, 4 as answer. The reason is the rate limiting. The question, see, this is the most prominent enzyme, most responsible. If you are asking me, I will tell that number one is the rate limiting enzyme of fatty acid synthesis that we have to take. So there is no option like that. So that is why I have taken 1, 2 and 4. If 2 only was there, then I would have taken 2 only. That is acetyl coenzyme carboxylase, because that is the rate limiting enzyme of fatty acid synthesis. One more point I will tell you. If the option has pyruvate dehydrogenase, will you choose that as answer? I will choose that as answer, because to divert the carbohydrate towards acetyl coenzyme so that there is no turning back. That is with the help of pyruvate dehydrogenase. That also determines a major role in making this glucose entering into fatty acid synthesis. So if you ask me the two prominent enzymes, number one position, I'll give pyruvate dehydrogenase, not even acetyl coenzyme carboxylase. But that was not there, so we are saved. So if pyruvate dehydrogenase and acetyl coenzyme carboxylase is given there, and if it is a single response type of question, I will choose pyruvate dehydrogenase. If pyruvate dehydrogenase is not there, and if this was a single response question, then I will choose acetyl coenzyme carboxylase. As it is a multiple response question and there is no option called as 2 only, I am taking 1, 2, 4 as the answer. So we'll go to the next question. This is a very simple question on G6PD deficiency. What will be the bilirubin finding? Actually, I won't say that it is bilirubin finding, but rather than I will say that what is the investigation finding. Okay, so multiple options are given here. So first and foremost, in glucose 6-phosphate dehydrogenase, what happens is there is decreased NADPH. If there is decreased NADPH, it is promoting or it favor hemolysis. So this is favoring a hemolytic jaundice. That is the first thing. Now, in hemolytic jaundice, what type of bilirubin is elevated? It is indirect bilirubin. So, indirect bilirubin should be increased. Direct bilirubin is normal. Then urine urobilinogen. Excess hemolysis means that excess urobilinogen is produced, because more and more urobilinogen, as more and more heme is catabolized, more and more heme is produced. So urine urobilinogen is increased, is classically seen in hemolytic jaundice. So what is the test for urine urobilinogen? It is Ehrlich test. So Ehrlich test is positive, highly positive here, then it is increased. So the answer here is option A. So direct is normal, indirect is increased, and urine urobilinogen is increased. So the next question is on glycine titration curve. Just before INI-CET, I have uploaded a video in YouTube, and the first question that I have discussed in that YouTube video was titration curve. So this is a very frequently asked previous year topic. 2 times this question came for INI-CET only. So that is very, very important. That is one thing that I want to tell you, titration curve. Now, in this titration curve, what is asked is Zwitter ionic pH. What is the pH at which any compound exists as Zwitter ion or ampholyte? It is the pH which is called as the isoelectric point. Right. Now, how can you calculate isoelectric point? Isoelectric point is calculated as the average of the ionization constants, like this. So average of 2 ionization constant, then we call it as the isoelectric point. So if in this question you can see that there is an ionization constant pK1 and pK2 given here, and question asked is find out the pi. So pK1 is 2.4 and this is 9.8. So that comes to 12.2, right? then average of it is 12.2 divided by 2. So it is 6.1. 6.1 we can take as 6. So 6 is the answer. Similarly, I will tell you one more thing. If this is glycine, there is no confusion. But if the question is aspartic acid, then aspartic acid has three ionizable group. One is the alpha amino group, then it has an alpha carboxylic acid group. In the variable side chain also there is a carboxylic acid group. So I am giving pK1 here, ionization constant of one ionizable group. This is pK2 here, right? Now for this extra carboxylic acid group, I am giving pK3. pK3. Now what is the isoelectric point? Is it average of pK1, pK2 and pK3? No. Then how you have to calculate Pi is Pi is equal to average of the ionization constant with isoionic species. What do you mean by isoionic species? That is, you should not consider that positively charged amino group here. You have to consider the carboxylic acid group, that is in the alpha carboxylic acid group, and the carboxylic acid group present in the variable side chain, which means that this is pK2 plus pK3 divided by 2. Okay, so as this is glycine, it is very easy. But if it was another amino acid, then you have to take the pK of the isoionic species. Then cyanide and carbon monoxide act on which hemoprotein? It's a very, very simple question. So cyanide and carbon monoxide, we know that it is acting on complex 4, and complex 4 is cytochrome C oxidase. So cytochrome C oxidase is the answer. Next question is, cancer cell consume more glucose by which of the following process? So cancer cell is a peculiar cell where you can see that more glucose is utilized. Why more glucose is utilized? Because whatever glucose is entering into a cancer cell is getting converted to lactate even in the presence of oxygen. So this is called as aerobic glycolysis only, even if the product is lactate. And as the product is lactate, we are getting only 2 ATPs. Okay, the question is not that why it is consuming more glucose. The exact reason of why it is consuming more glucose is it produces only 2 ATPs. Why it produces only 2 ATPs? Because the glucose is converted to lactate even in the presence of intact oxidative phosphorylation and intact oxygen and intact mitochondrial pathways, it is converted to lactate. So there is a deficiency of ATP there. So it takes up more and more glucose. So what is the answer here? It is increased lactate production even in the presence of oxygen. Okay, so as it is a straightforward question, you can answer it like increased lactate production even in the presence of oxygen. It is not glucose transporter 2. Glucose transporter 2 is not the one which is present in all cancer cells. It is specifically present in beta cells of pancreas, liver, certain sites only. So that is not the answer. Increased fatty acid oxidation, no. Increased conversion of pyruvate to acetyl coenzyme, never. That happens in a normal cell. So this is from this option, C is the answer. If that is not there, if there is something like metabolic reprogramming or presence of an isoform of pyruvate kinase called as PKM2, this is the exact reason whereby cancer cell glucose is converted to lactate. It is called as metabolic reprogramming. Metabolic reprogramming. Okay, so the old theory was Warburg hypothesis. That is replaced by the new theory which is called as metabolic reprogramming. So that is presence of an isoform of pyruvate kinase called as PKM2. Usually what is present is PKM1. In cancer cell what is present is a dimeric form which is called as PKM2. Next question is a 30-year-old female with a strong family history of breast and ovarian cancer referred for genetic testing. What is a single best genetic test to diagnose this condition? So in a usually that mutation is BRCA 1 and 2. Usually familial breast cancer, definitely BRCA 1 and 2 is the most common gene that we have to study, right? Now, how will you find out the genetics? First and foremost, we should understand what is the type of mutation here. So, based on the mutation, we have to analyze regarding what technique that we have to use. In the case of BRCA1 and 2, it is usually a mutation which is maybe a point mutation or some or small mutation. It is not something which affect the length or something which affect the gene dosage. It is not something which is affecting gene dosage or affecting the length. Mostly it is one or the other kind of point mutation. Right. So if it is a point mutation, then we have to do first a PCR. Then what are the techniques that we can use? A restriction fragment length polymorphism or a Sanger sequencing can be used, or a latest technique like next generation sequencing can be used. Next generation sequencing include whole genome sequencing, whole exome sequencing, like that. So now we will see the options. qPCR. See, qPCR is something called as real-time PCR. Real-time PCR is used when we are doubting a mutation which affect the length. For example, trinucleotide repeat mutation. That is something which affect the length. Then if we are doubting such kind of mutation, a large gene deletion, for example, CFTR gene, a gene is deleted, such kind of things, then a gene dosage is affected, then only we'll do quantitative PCR. Or MLPA can be used, then amplicon length analysis can be used, like that. So these are the mutation detection technique. So based on what is the most predominant mutation, the technique also vary. So first option is qPCR for quantification. No, it is not something usually causing a length mutation. It is, so that is not the answer. Exome sequencing, it is coming under whole, it is coming under next generation sequencing. So exome sequencing is the best method. RT-PCR, which detects the RNA level. Here the question is DNA. BRCA gene genetic testing is for BRCA 1 and 2. They are genes, they are DNA. So in a DNA we will not do a reverse transcriptase PCR, which is doing that is for what you called as the gene expression study. It is not asking gene expression, it is asking gene mutation. So definitely that is not the answer. Southern blotting for DNA replication. First thing, Southern blotting is not something for DNA replication, but Southern blotting is based on DNA hybridization. So from that DNA replication, that is a wrong option. Now what about Southern blotting? Can we use? Yes, Southern blotting can be used. But the question here is single best genetic test. That should be a next generation sequencing, which include whole genome sequencing or whole exome sequencing. What is the difference between genome and exome? Genome means the entire genome is sequenced. It is very costly. But if we are less, like if we have resource of funding or resource is less or money is less for us to do a testing, then what we can do is we can just check only the coding regions, which means only exons are sequenced. Exons regions are sequenced. All the exons are sequenced. That is called whole exome sequencing. Then our area where we have to sequence can be reduced. So whole genome sequencing is very costly. So if we want to reduce the amount, what we can do is whole exome sequencing. So in this, the answer here is exome sequencing. The next question is a 12-year-old boy brought with rigidity and cognitive impairment. So image is shown here, an MRI image is shown here. It has tiger eye sign. And what is it? So this is not directly a biochemistry topic, but there is something related to biochemistry here. That is why I have chosen this question to biochemistry. So this is a defect in the utilization of pantothenate, that is vitamin B5, pantothenic acid. So this deficiency is PKA, that is pantothenate kinase deficiency, which is associated with vitamin B5. So its radiological aspect, definitely radiology faculty will be telling you. Let me tell you about this PKAN deficiency. It is actually a neurodegenerative disorder. which is otherwise called as Halowarden-Spatz syndrome. So this is a neurodegenerative disease where there is chorea, dystonia, then Parkinson's features, all these are there. Plus radiological finding there is eye of tiger sign. Okay, so I just want to discuss the biochemistry part here. It is something related to vitamin B5 or pantothenic acid. The next question is, an uncontrolled diabetes, in which of the following cell there is decreased glucose uptake? Various ways this insulin dependent glucose transporters are asked. This is last time it was asked like after exercise, in which of the organs the glucose transporter is elevated. So all these questions are pointing towards insulin dependent glucose transporter. Where is insulin dependent glucose transporter present? In the heart, in the skeletal muscle, and in the adipose tissue, right? So, if there is insulin deficiency, glucose uptake is reducing in an insulin dependent glucose transporter location, and that should be skeletal muscle. Okay, so that is answer is B. So the next question is, which method is used to identify different mutated strains of the same species of Mycobacterium tuberculosis? So before going to the question analysis, let me tell you one thing, that if two strains of a same species is produced, or then what is being changed? It is the genetic material is being mutated. So in a Mycobacterium tuberculosis, what is the genetic material? DNA is the genetic material, which means that this is a change in the sequence or a mutation in the DNA of the Mycobacterium tuberculosis. So we have to find out something which sequence the DNA in a case of Mycobacterium tuberculosis. That is the clue here. Various techniques are given here. We'll see one by one. So what are the techniques that are used for genotyping the strains of bacteria, especially Mycobacterium, because it is a DNA material? So generally what we call for all the materials together is called as, or all the techniques together is chromosome mutation analysis. That was one of the option. Then what is given as RFLP, that is restriction fragment length polymorphism, where two strains are identified using an enzyme called as restriction enzyme. That is called restriction fragment length polymorphism because there are unique restriction sites in each of the strain, or if the mutation is in a particular restriction site for a particular restriction enzyme, then the band produced by these two strains will be differing. So this is a very old, at the same time widely popular, or one of the popular and easily available technique that we can do is RFLP. Moreover, there are other techniques. So another technique is spoligotyping. Spoligotyping means we should know something about this DNA material of a Mycobacterium tuberculosis. So in this, there are certain regions which are called as direct repeats. Okay, direct repeats are there, and in between or on either side of the direct repeat, there is a unique spacer. So direct repeats are not unique, but on either side, there are certain unique spacer sequence. So if we are targeting on this unique spacer sequence, which is on either side of this direct repeat, and that is called as spoligotyping. Now, is it a best method? No. There are even more better methods are there. That is Miro. What is that? It is multiple interspersed repetitive unit typing. It is like directly typing the variable number tandem repeat. The same we are doing in restriction fragment length polymorphism also, but this is an old technique. We are not directly sequencing. Here we are directly sequencing the variable number tandem repeats. So the variable number tandem repeats are typed here. In spoligotyping, the spacer sequence on either side of the direct repeats are like analyzed. Here it is V and TR are directly analyzed. So it is much more higher discrimination quality and much better technique than spoligotyping. Then the final word for genotyping of strains is next generation sequencing, where we can do whole genome sequencing, where we can find out the different strains of the bacteria. Okay, so now we'll come back to the question. Which are the methods? It's a multiple response, RFLP, yes. Chromosome mutation, yes, everything is coming under that only. Then polygotyping, yes, and Miru VNTR, yes. So the answer here is 1, 2, 3, and 4. So the hardest question INI-CET, if you ask me, definitely this plasmon resonance question, which is SPR graph was given. Very unlikely, and I will say that it should not have been asked in an exam like this. So definitely, but once the question came, you should be aware of that such question can come. Okay. And definitely we should remember in a competitive exam, definitely such a difficulty level questions will be there. Definitely it should be there. Okay, so come back to this question. What we can see is it competitive ELISA? So two options were competitive ELISA, two options were surface plasmon resonance. What we should understand is that ELISA also we are detecting antigen and antibody complex. Immune complexes are being detected. But what is the difference in ELISA? In ELISA, different ways we can do competitive ELISA. Different ELISA techniques are there, but it is like a conventional PCR. What do you mean by conventional PCR? We'll do the amplification and later on we are analyzing. Likewise, ELISA is a technique where we are doing the antigen antibody reaction, and finally we are doing a reaction whereby we are detecting the presence of antigen or antibody. That is what we are doing in ELISA. So that is afterwards. But in this graph, what we are checking is a real time. Real time means as and when. For example, real time PCR, we know as and when amplification take place. Likewise, as and when antigen antibody reaction is taking place, we are checking at a particular physical property, and that is called an angle, which is called as, I'll show this angle, surface plasmon resonance angle. So if you see, it is a very complicated mechanism, no need to learn the physics of it, but basically what you should understand is there is a light source. So once the antigen antibody complex is formed, it produces a set of waves which are called as plasmon waves. Now, once the plasmon wave starts to appear, then we can measure what is called as a surface plasmon resonance angle. Okay, so now you see this picture. So initially, when there is no antigen antibody complex formed, there is no surface plasmon resonance. Why? Because surface plasmon resonance happens only if the antigen antibody complex is formed. Okay, so initially there is no response. Now when we start to add antigen, gradually you can see there is an ascent. So this is like an association of antigen antibody. When a peak, you can see there is a particular area where it's almost plateauing, and see this is the formation of the complexes. Then what we can do is, then the antigen and antibody complex starts to detach. So dissociation. So what we are measuring is the association of antigen antibody and dissociation of antigen antibody. That is what we are measuring. But how we are measuring, that is based on that plasmon wave that is produced when antigen antibody complex are formed. So we are adding antibodies, and when the antigen antibody complex is formed, the surface plasmon resonance starts to rise. Then once it dissociates, then it will regress. So this is called SPR, surface plasmon resonance curve. That's the first thing. Okay. Now, what is measuring? Then again, so if you understand that this is not ELISA, you understand that this is a real-time method, it is not ELISA. Okay. So you cut down these two options. Then again, surface plasmon resonance, two options are there. Antibody concentration or antibody affinity. This is one of the gold standard method to detect antibody affinity. So we are like a monoclonal antibodies if we are making. So if we want to know whether this monoclonal antibody has affinity towards the antigen that we are looking for, then we have to check its affinity status. So if we want to check the affinity status, we'll do this real-time surface plasmon resonance, and we'll check for this surface plasmon resonance angle, and there is a detector to detect that. So do you understand that? So it is not for antibody concentration. We are not measuring antibody concentration from the beginning onwards, it is there. So whether it is associated or dissociated, antibody is there. Whether it join or bind with the antigen is what we are searching for. So once the antigen antibody bind, then surface plasmon wave starts to appear, and surface plasmon resonance curve will show an ascent, and then later it stops. So it's a real time measurement. It is a label free technique. What do you mean by a label free technique? In ELISA, what we'll do an antigen antibody complex, and finally we'll add a label for what? For detecting the presence of this antigen antibody complex. That is, we are using a label is in there. But in surface plasmon resonance, two advantages are there. One is it is real time. It is a real-time surface plasmon resonance, and second, it is label-free, unlike an ELISA technique. And this is considered as a gold standard antibody affinity detection method. I hope that is clear. So with that, we conclude the discussion of INI-CET. So this is what I want to say in biochemistry part. It is mostly moderate to difficult, but don't get disheartened by this. Life has to move on. That is what I said in the beginning. Let's go, grow, and move on. Don't stumble over what is behind you. And all the best for you. Thank you.